And thank you, Dr. Sorenson and Chris, for coming for our Community Science Saturday, where we're learning about Firefly Watch. Thank you so much. Turning it over to you now.
All right. So I'm going to go ahead and share my screen. And hopefully this will all work right. Let's see here. Okay. How about that?
Well, nope. Now we're seeing your
now you see the wrong one.
Yeah, we tried
it. Wait a minute. And just let's try it again. And then let me see if I can swap it. And let's see here. Now, which one are you seeing?
I'm still seeing. Oh, now we're good. Now we're good.
Now you're good. Okay. So you only have one picture of
that's right.
A large firefly, and it says welcome. So we're good to go. Hopefully.
It is.
Right?
One thing I forgot. Yes, and one thing I forgot is questions are highly encouraged, but let's put them in the chat box, and then I'll act as the kind of curator for questions, so we're not bombarding the presenters. And now it's up to you.
All right. So let's see if we can actually advance. There we go. My name's Clyde Sorenson. I'm the one with the beard. And I'm from North Carolina State University, from the Department of Entomology and Plant Pathology. And my co-presenter, who's going to be talking to you shortly, is Chris. And we're going to kind of, so we're neither one of us is from Maryland, but we do have you kind of wedged in between us, between North Carolina and Massachusetts. And so hopefully, a lot of what we say is going to be significant for y'all in your mid-Atlantic position between our two states. So to get started, I'll show you real quickly a little outline of what we're going to try and do. I'm going to first talk about fireflies and their biology, kind of with a North Carolina slant. And then Chris is going to talk to you about fireflies from a more northern perspective. And he's also going to spend a fair amount of time with you talking about the Firefly Watch program. And so I'm going to go ahead and jump right on in and start talking about fireflies as it relates primarily to North Carolina. But I want you all to remember, and this is really important, a lot of the players we have in North Carolina are in Maryland. And when Chris gets around to talking with you, a lot of the players in his area are also in Maryland. So what we're going to talk about, I think, hopefully is going to apply for everybody. So let's get on it. We've been talking a whole lot about who calls what, and some folks say firefly, some folks say lightning bug. I grew up primarily in the South, and it was primarily lightning bugs. And by the way, when y'all were talking earlier about using lightning bugs to make bioluminescent jewelry, and you thought that was kind of disgusting, that's not nearly as bad as the game my brothers and I used to play. Well, it was mostly me trying to convince my brothers to do it, and we called it tracer boogers, which was you take a lightning bug and put it up your nose and then go like that to make a glowing projectile. Anyhow, enough of that. What is a firefly? Well, fireflies aren't flies, actually, and they're not bugs. They're actually beetles. And the ones that we're concerned about, that we're going to spend most of our time talking about, belong to the family Lampyridae. And Lampyridae means light bearer, which is very appropriate, I would say. To the ancient Greeks, they were Chysolampus, which basically translates into the one with the glowing butt. So that's kind of another good way to remember who they are. They are beetles. They're closely related to several other families of beetles that also incorporate or include bioluminescent members. The Fingodidae, which we'll talk very briefly about a little bit later, also bioluminescent. In fact, some of the click beetles, which lampyrids are kind of closely related to, are also bioluminescent. In fact, the brightest, excuse me, let me close my door. That's my chocolate Labrador welcoming committee downstairs. Apparently somebody came to the door. But the brightest, the most intensely bioluminescent insect in the world is actually a click beetle that has two very bright glowing spots on its pronotum that look like headlights, which is just really cool. Anyhow, beyond the beetles that are bioluminescent, there are some other bioluminescent insects. We'll talk very briefly about a couple of them a bit later as well. But it's important to remember that fireflies are actually beetles. And compared to many other beetles, they're sort of soft body. They have sort of soft forewings. Some important pieces and parts of fireflies that you should be familiar with because they help you identify them are identified in this figure here. So the nature of the antenna sometimes can be important in helping you sort out who it is you're looking at. The size of the compound eyes that fireflies have, in many cases, will let you tell which sex they are, because in most species of fireflies, the males have larger compound eyes than the females. This structure here on the immediately behind the head is called the pronotum. And this is actually the first thoracic segment, the top plate of the first thoracic segment of a beetle's thorax. And in fireflies, the pronotum, as it turns out, is really, really important because the shape and the color and the markings on the pronotum a lot of time are crucial to understanding exactly which species it is you're looking at. And then another small structure that is useful is the scutellum, which is another one of the top plates in the thorax. It's just a little triangular thing right here. The color and shape of that a lot of times is useful in identifying a firefly. And then the forewings of a firefly are called the elytra, the singular elytron. And again, the color and the markings on the elytra a lot of times will help you identify who it is you're looking at. In some fireflies, there's a structure called an elytral fold, which is kind of a little longitudinal wrinkle. And the shape and the color that might be associated with that a lot of times can help you identify a firefly as well. But fireflies are fairly distinctive insects. In most species, this pronotal shield kind of hides the head when you look at them from above. And that's important because we're going to see in a minute that there are lots of other insects that want to look like a firefly. And that'll help you separate them from true fireflies. All right, when we look at the number of species of fireflies in the world, there are probably a couple thousand, which sounds like a lot. But given that there are about 365,000 species of identified beetles, they actually constitute a very small fraction of the total diversity of beetles. But that's still a lot. In the United States, we think we have about 170 species or so in 20 or so genera. And I say about, and we think, and or so, because we don't know. We actually don't know how many species we have. this is a situation that's in a tremendous amount of flux. Fireflies, compared to some other beetles, are really sometimes hard to sort out because in many cases they don't look all that much different, but they act a lot different, and that's really weird in the insect world. We have about 170 species. It's almost certainly more than that, though. In North Carolina, there's somewhere between 30 and 50 species. I recently put together a catalog of the known species of fireflies in North Carolina, and it came out real close to 40. And I am certain that's an underestimate of the species in North Carolina. In Maryland, I suspect you have at least 30 or 40 species as well, probably more. Now, when we talk about the groups of fireflies, the genera of fireflies that are prominent, most of the fireflies that people encounter and that they notice belong to about four genera. Probably the most familiar to most folks is Photinus. That's a Photinus firefly right there. That's probably actually the most widespread and familiar firefly in most of North America and Eastern North America. Anyhow, that's Photinus pyralis. The Photinus fireflies typically have a shield that's got some yellow and red markings and usually some sort of a central mark. Most of them have dark colored elytra with well marked bands. They tend to be, when you look at them from the side, sort of kind of flattened profile. Photinus is very, very widely distributed in Eastern North America. Next genus that's widely distributed in Eastern North America is Photuris. The Photuris fireflies are actually, for those of us who have messed with them for any length of time, extraordinarily annoying because they're really behaviorally plastic and lots of them look very, very similar. Most Photuris fireflies have on their pronotal shield a central mark with lateral red spots, lots of them. And when you look at them from the side, they tend to be kind of hunchbacked. Their prothorax and the rest of their body kind of assume an angle, and they have a very well developed elytral fold, as I was talking about. And lots of them have stripes on their elytra. A lot of the Photuris have really long legs, and they tend to be, many of them tend to be kind of large for fireflies, and we're going to talk about why that is in a little bit. The third genus that's pretty widely spread and very, very noticeable, Pyractomina. In many places, these are the earliest fireflies, the earliest glowing fireflies, I should say that you see in the spring. A lot of these tend to be large. And one of the things that sort of distinguishes them is that a lot of them have on their pronotal shield, they have these dark marks on the lateral margins. And you'll notice their pronotum tends to be shaped a little bit more differently from the other two genera that we've talked about. These are really cool. I should add, there is some difference there, on average, some difference in the color of the light that the different genera produce. And Pyractomina tend to produce an orange or reddish flash, which is kind of exceptional and noticeable. And then the fourth genus of glowing fireflies that many people notice and pay attention to is Phausis, or the ghost fireflies. That's actually an advertisement for a brewery up in Asheville. The blue ghosts, the ghost fireflies, are very small compared to the others. They're very dark. Most of them, in fact, all of them, as far as I know, have females that are wingless and larviform. And what's exceptional about the ghost fireflies is that, especially the one that's most widely known, Phausis reticulata, the quote unquote the blue ghost, has a behavior where it doesn't flash but it flies around with its light on, sometimes for as long as a minute, leaving a little blue-green path through the forest. And when you see hundreds of these doing this, it's really extraordinary. And I always like to imagine what it might have been like to be one of those Scottish settlers coming into the Appalachian Mountains back 250 years ago. And, you know, a lot of folks from Scotland are fairly superstitious, or were back then. And to see all these little fairies wandering through the woods, and when you get close, they disappear, must have really freaked them out. They're called blue ghosts, but their lights are actually green. They just look blue because of a weird effect called the Purkinje effect. I can't say that very well. But basically what that means is if you look at a faint green light at a distance, it blue shifts. And that actually happens with some other fireflies as well. So those are the 4 most widely encountered genera, but they're not the only genera that produce light. There are a couple others. And it's important to note that widely in lampreyrids, even if the adults are dark, a lot of times the larvae are not, which is kind of cool. Now, not all fireflies are lightning bugs. About a quarter of the lampreyrids don't have light organs as adults, and so they're day flying, and they don't glow, and they still, though, look more or less like fireflies. And so most of these, and this is a good example, one that's actually quite active right now in parts of North Carolina. This is Lucidota atra. Most of these we think rely primarily on pheromones for sexual communication. And it's kind of an interesting story because we think some of these day flying dark fireflies actually evolved from night flying lit fireflies, but they went back to avoid nighttime predators. So it gets really, really weird evolutionarily. Fireflies have lots of really cool evolutionary stories to tell us, and we don't really know all of them yet. But it's important to remember that not all fireflies are actually lightning bugs, and that, real importantly, not all fireflies are even fireflies. There are a lot of insects that like to mimic fireflies because many fireflies, particularly those in the genus Photinus, are protected by noxious chemicals called lucibufigins, which are chemically related to the toxins that toads produce, even though they're not of the same origin. And so you have things like soldier beetles that want to look like fireflies, and cerambycid longhorn beetles that want to look like fireflies, and other beetles that want to look like fireflies. And then, for that matter, there are some other insects that aren't beetles that want to sort of look like fireflies, because if you eat some fireflies, you come to regret it very quickly. In fact, I had one of my graduate students a couple of years ago on a dare put a firefly in his mouth, and he thought it was worth $4, and his mouth was numb for about 45 minutes after he did that. So maybe you don't want to do that. All right, so of course everybody is interested in fireflies, lightning bugs, because they make light, and that's kind of an interesting process in its own right, because almost all the energy that goes into firefly light production comes out as light. They waste very little of the energy that goes into this system in the production of waste heat. And all the ways we have come up with as humans to make light waste energy as heat. So the production of light in fireflies relies on a chemical called luciferin. All right, why aren't we moving? Okay, apparently I moved my cursor in the wrong place. So luciferin produces light if it's combined with some other materials. And so those other important components are a real important enzyme called luciferase, oxygen, and the cellular fuel that all cells use to run, and that's ATP. And so when these elements are combined in the right proportions, you get light. And again, almost all of the energy that goes into this comes out as light, which is really, really cool. Now, I said earlier that there are other insects, other beetles and other insects that produce light. Some that you should be familiar with because you might encounter them would include the glowworms in the family Fingodidae. The Fingodids are bizarre beetles because for one thing, they don't look much like beetles. This is an adult female Fingodid glowworm. That's what she looks like in daylight. That's what she looks like if you take a time-lapse image and combine it with a flash image, and that's what she looks like just in the dark. And the Fingodids, the females and the larvae, have light organs on their body segments that they keep lit while they're moving through the forest floor. And so consequently, they look like a little subway train. And so lots of folks like to call them railroad worms. Most of them feed on millipedes, and I think some of them feed on snails. And so sometimes you can find them inside the empty husk of a millipede that they've eaten. As I said, the females are what we call larviform, as you saw in that first picture. That's what the male looks like. And so it's always been kind of a puzzle to me how something that looks like that's about 1/3 the length of the adult female, manages to successfully mate with the female. But apparently they've been doing it for like several million years, so I guess it works. Another group of bioluminescent insects includes some groups of flies, true flies. Probably one of the most famous of these is the glowworms that we find in Australia and New Zealand, more prominently in New Zealand. These are the larvae of a fungus gnat, and they make a blue light, the larvae do, and they live on the ceilings of caves and grottos, and they basically use that light to attract prey into their webs, and then they reel their captured prey up and eat it. We in North America have a glowworm fly. It's called Orphelia, and it may produce the bluest biologically produced light of any insect, maybe of any animal on the face of the planet. And you can find these things sporadically in little overhangs and ditch banks and places of that sort. And again, we think they use that light primarily to attract prey. Okay, so does anybody have any questions at this moment? Or are there any questions in the queue that we need to address?
No questions thus far.
All right, if not, then we'll go ahead and we'll get back to fireflies. We'll talk a little bit about their biology and what they do. Most fireflies are predators as larvae, and in fact, they're very vigorous predators. It's really kind of phenomenal how they go about it. But little tiny firefly larvae can kill great big earthworms and snails and slugs. They're venomous.
Clyde, you just froze on us. Ooh. Clyde just froze on us. Did he pop off? No. There, he popped off. He's probably going to pop back on in a second. We're all used to this.
Well, you can certainly, I don't know if any other questions have come up since that question prompt. I would be happy to answer them while we're waiting for Clyde to hop on if there are any.
I don't see any, but anybody have a question? That was a very bizarre looking male glowworm. I can't get that out of my head. Laura wants to know any Phausis in Maryland.
That's a really good question. It's something that Clyde and I were just talking about yesterday, because I sort of said probably not, but what his point was, because we don't have Phausis at all up here in Massachusetts or anywhere in New England, of that particular kind, certainly. And it looks like Clyde may be back, but I'll just finish the answer that what we discussed, which is his point was that as in North Carolina, as he's begun and people have begun to look for it more widely, it's sort of, once you take the time to look for it, you find it places that you wouldn't, that hadn't been seen before. It's because it's active for a very short period of time, and it's not particularly, widespread or conspicuous even. And so you can, he's been surprised often to find it places, and that struck us and struck him in particular that, well, people should look for it in Maryland because you never know. So that was what we were discussing yesterday. But let's see if maybe we've got audio back from Clyde.
Clyde, are you back with us?
Not quite. So guess we can, again, if anyone else wants to put a question in there.
Now we're going to do this right.
Great.
Okay.
Good.
Now you can hear me. Zoom, you gotta love it. Just gotta love it. It's what a wonderful invention. Okay. Are you looking at the right thing again? Okay. So to get to the Phausis point, so Jerry Reynolds can back me up on this. We had learned about a population of what might be a new species of Phausis in the Piedmont of North Carolina. And there was only one known population. So this year I kind of rallied the troops on Facebook and I said, I want y'all to look in your backyards. And very shortly, back in late April, early May, we found 3 more populations. And then Jerry asked me, he lives not far from where I do in Johnston County. He said, what do you think it's worth me looking? And I said, well, sure. And he went out and made a kind of half-hearted look one time and said, no, I didn't find any. And then I said, oh, okay. And he said to himself, maybe I better actually go look. And he went out and looked, and another night, and lo and behold, he had him in his backyard. And he told me, I have him in my backyard. And then it occurred to me, maybe I have them in my backyard. And I went out, and be doggone, I have him in my backyard as well. And I've been living in this house for 25 years and did not know it until this year that I had a ghost firefly living in my backyard, and that it's looking more and more to me like a new species that hadn't been described by science yet. So to get to that very long answer to a very short question, it's worth looking because you don't know.
Yep, and that's what Laura just asked. What do we look for if we're out there? And that's part of the community science stuff. If there are discoveries made all the time in your own backyard.
That's right. So the Phausis fireflies typically fly after it's solid dark. The Phausis reticulata typically flies in the mountains of North Carolina in early to mid-May, and then again in July, or whatever the heck it is we're dealing with, we saw in late April and early May, and then it persisted into quite late into May. We don't know if it's going to have another flight. They're very faint. You have to look deliberately for them, but once you see them, it kind of blows you away that you hadn't noticed them before. So yeah, I don't have any idea what to expect. If they're, to my mind, if they're in the southern Appalachians of North Carolina and in the foothills of North Carolina, it would surprise me if there weren't Phausis, perhaps at least in western Maryland, where you get a little tiny taste of Appalachian habitat. So it's always worth looking. And the worst that happens, you don't find it, but you see other cool stuff. So maybe you find a glowworm. We found lots of glowworms looking for Phausis. So, all right. So where did y'all lose track of me? I was talking about how they're vigorous predators, I believe, as larvae. And they feed primarily on other invertebrates. A lot of them feed on earthworms, on snails, on slugs, things like that. Many fireflies are habitat specialists, and you'll find the larvae only in particular habitats. Many fireflies have wingless adult females, and so they also are kind of habitat specialists. To that end, because a lot of them rely on mature hardwood forests or native forests, not necessarily hardwoods, exotic plants can sometimes perhaps interfere with their ability to communicate. We'll talk maybe a little bit more about that a little bit later. Again, many of them are wingless as adults, and most of them, with a couple exceptions, don't feed as adults. This is what a lot of firefly larvae look like. And again, most larvae are bioluminescent. In fact, the eggs of most fireflies glow, and the pupae of most fireflies glow as well. And we think they actually evolved this bioluminescence initially to warn predators not to eat them because they're nasty, and that it was later co-opted by the adults for sexual communication, which is what we're going to talk about now. And in most fireflies, they use their light primarily as adults for sexual communication. Again, different genera often have different light colors. Each species has its own signaling system. And so consequently, in most cases, or many cases, you can identify fireflies by the signals that they make, because they are species, more or less species specific. In most of our species in this part of the world, the males fly around doing the risky behavior of advertising and signaling. They'll make their characteristic signals. The females typically are either on the ground or in low vegetation, or sometimes high in the treetops, perched, looking for the males. And so when the males make appropriate signal, female sees the appropriate signal. If he's doing it just right, she'll respond back with her own species specific signal. And then they kind of call and response, signal back and forth to each other, the male approaching the female, until they get close enough for him to run over and shake hands, and then nobody flashes anything for a while because they're doing something different. And so this is a real short video of Photinus pyralis, the Big Dipper, and this is the consummation. And what we have here is a very frustrated male trying to horn in on the activity. Well, we already have a paired male and female. And you'll notice there's a third male in the background, still trying to figure out what's going on here as well. So each species has its own signaling pattern. Get on to the next slide. As I said, Photinus pyralis is this very common one through much of Eastern North America. If you watch on this video as it rolls, there he is. We call him the Big Dipper because they make a U-shaped flash every couple of seconds. He's getting closer to the camera as he comes in. You'll see he makes that swooping U-shaped flash. This is a species that also tends to fly right at dusk, and so you can actually see the firefly before it gets dark. He makes that signal. The females are watching. He'll flash. You count 1, 2. She'll make a half-second flash back. He'll see that, make his flash again, and so back and forth until they get together. That's Photinus pyralis. This is Photinus consanguineus. We sometimes call him the double tap. You'll have to watch very carefully on this video, but you'll see a firefly making 2 flashes about a half-second apart. And again. And it should be a third sequence there. And so the females in this species are watching for that double flash. There's the consanguineus right there. Consanguineus is much smaller than pyralis. And you notice it's got a dark bar that goes all the way across its pronotum. That's a kind of a hallmark for it. And then here's a third one. This is Photuris frontalis. This is one of the synchronous fireflies. And let's see what you'll see is just a steady train of flashes going across the screen. If you watch carefully, they're pretty faint, unfortunately, in this video. Yeah, there he was. Flash, flash, flash. And so each species has its own signaling system. And in many of the species, that's a fairly fixed pattern. There are, unfortunately, when you're trying to identify them, exceptions. In most of the Photinus species, the male and female flash patterns are fairly well signaled or established rather, but in particularly in the Photuris, single species may have multiple flash patterns, which makes it difficult to try and sort them out sometimes. And then some species use their flashes for, well, I'll just say, at least in one case, a more nefarious purpose, and we'll talk about that in a minute. Now, lots of fireflies, if you caught them as a kid, you probably noticed they have kind of a funky smell to them. That may be related to the fact that they produce some toxic chemicals that we call lucibufficants. And as I said earlier, these are related chemically to the toxins that some toads produce. They're not only toxic, but they're extremely distasteful. And that's why we see so many other insects mimicking them. And that also may have something that, well, it has a lot to do with what I'm going to talk about in just a second. There are possibly other chemicals involved. As it turns out, lucibufficants are not terribly volatile. So we're probably not smelling lucibufficants, but we're probably smelling other chemicals that they produce that not only, along with the flash, not only help protect them, but also probably help warn predators that you don't want to eat them. Lots of fireflies that produce these chemicals or acquire these chemicals do what we call reflexive bleeding to help kind of protect themselves. So when you aggravate them, they spontaneously leak blood with the chemicals out. And then any predator that's grabbed them is going to be confronted by this nasty toxic smell or toxic substance on the exterior of the firefly. I actually watched a toad one time eat a Photinus, And you could actually see the Photinus glowing through the toad's throat. And then shortly, the toad yacked it back up and sat there wiping his face with his feet while the firefly, gooey in toad gut, walked off completely unharmed. And so it's very effective. As I said, we know graduate students probably shouldn't put them in their mouths either. So I've kind of alluded to this several times, but there are some fireflies that do different things. As it turns out, most Photuris fireflies don't manufacture lucibufficants. They don't make their own toxins. And as it also turns out, some of the adult female Photuris are predatory. And what they're predatory on primarily is the males of Photinus fireflies. So in some of these Photuris species, the virgin females will sit around watching for a male of their own species, and they'll signal to their paramour appropriately, and they'll get together and they'll mate, and all things are good. But after they've mated, on subsequent nights, they watch for Photinus males, and they mimic Photinus female flashes, so that when that male comes down, thinking he's going to get lucky with a Photinus female and runs over to shake hands, she lashes out, grabs him, and then eats him. And she's basically harvesting the lucibufficans from that male Photinus that she then uses to protect herself to some degree, but also to protect her eggs and herself, primarily from spider predators, we think. And these femme fatale Photuris are very aggressive predators. I took that picture right there, by the way, at my back porch light. That's a poor pyralis going down the gullet of a versicolor complex female Photuris. But you'll actually see them run them down in midair. They'll hunt them down in midair, and they'll steal them from spider webs. They really want those protective chemicals. Now, before I move on, maybe even pass the torch to Chris, there are a couple other phenomena that you should be aware of that are kind of noteworthy. One of these is the synchronous fireflies that have become sort of noteworthy. In North America, we have two species that have become widely known for their synchronous behavior. One of these is Photinus carolinus, which is pictured here. This is a species that's found in the Appalachians from probably northern Georgia, at least into Pennsylvania. Again, it wouldn't surprise me in Western Maryland if there were not populations of synchronous Photinus carolinus. What's kind of noteworthy about these guys is that the males have a flash train signal of somewhere between 8 and 10 flashes, and then they go dark for 4 or 5 seconds. And when you have high enough populations, all the males will flash in synchrony and go dark in synchrony. And when you have several hundred or even a couple thousand of these things all doing it together, it's a pretty awe-inspiring event to witness. The other species that we have that's kind of become notable for synchrony is the frontalis that I showed you just a little bit ago, Photuris frontalis. This is a species that's more associated with lowland, wetland habitats like cypress forests and mesic or wet forests, hardwood forests as well. I actually have these guys in my backyard, and these guys are really cool because they synchronize their individual flashes. So they'll pretty much continuously flash at about a half second or 3/4 second interval. And all of them that can see each other will synchronize their flashes. So everybody's flashing together. And again, when you have very many of them, it's pretty incredible. I only have populations that get up to 45 or 50, but seeing 40 or 5 or 50 fireflies that are all flashing together. Kind of cool. In the Congaree National Park, they have thousands that are doing that. And so really, really, really cool. All right, so I'm going to stop at that point for a second to see if there are any other questions. I could continue and talk a little bit about habitat, but I think Chris is going to be talking about some of those same kinds of things as well. So are there any questions that we need to tackle right at the moment?
Yeah, Clyde, Jerry wanted to know other non-toxic insects that mimic fireflies with light to avoid predation.
Say that again.
Do some other non-toxic insects mimic the fireflies with light to avoid predation?
I'm not aware of any that do that. I suspect some fireflies might, in some parts of the world, might mimic each other, and there may be at least Mullerian mimicry going on. But I'm not aware of, Chris, are you aware of any other bioluminescent insects that mimic fireflies?
No, I'm not. And but I do think, you raised an important point about fireflies themselves. You've already mentioned that, these Photuris that don't have lucibuffogens, and mentioned it in the context of their behavior to acquire lucibuffogens by eating Photinus fireflies. But similarly, we could see their flash behavior as behavior that has also evolved in part as a mimicry of those chemically defended Photinus as well.
Right. So when we talk about mimicry, there's many different kinds of mimicry, but the two most widely recognized are Batesian mimics, where you have a non-toxic mimic mimicking a toxic model. And so Photuris and Photinus would fall into that category. And then Mullerian mimicry happens when both members of a mimicry syndrome are toxic. And so Photinus flashing, different species of Photinus flashing are kind of reinforcing that signal to predators that, anybody that's flashing probably just isn't good to eat for at least some predators, right? Some predators don't care, by the way. Some predators don't give a rat's patootie if you are toxic. They're going to go ahead and eat you anyhow. And that's true with some spiders, and we could go off on a whole lot of tangents in that direction. But I'm not aware of any non-beetle insects that use light to mimic fireflies. I'm not aware of that.
Yeah, I guess if you were to step back from our adult fireflies into the larval fireflies, and the variety, as you said, of different larval beetles that are, for instance, in a forest understory, I don't think it's been looked at specifically the extent to which there's mimicry going on. But when you look at things like the Phengodidae, and then you look at firefly larvae, that is a fascinating question, the extent to which those signals being produced by larvae are reinforcing each other through either Batesian or Mullerian mimicry. So that is an interesting possibility.
So how are we doing on time? Is it probably a good time for me to pass the torch off to Chris? I have some other things to talk about, but we could just come back to them later.
I think it's a good time to pass it off to Chris, and we can always come back.
Very good. All right, so I'm going to stop sharing. Hopefully I won't disappear altogether. And I'll let Chris share his screen then.
Okay, so hopefully that is working now for me me sharing my screen. All right, great. Well, thanks, Clyde. Clyde has done a great job of setting up so much of the fascinating biology and diversity of fireflies that are out there. And I'm going to really try to dive into what You might choose to do what we hope you will choose to do in terms of watching fireflies in your own backyard, your neighborhood, or even if you have to travel a little further afield to find them. That's what we're encouraging you to do through Firefly Watch. And so we'll talk a little bit about the challenges of that, how you get involved, and sort of wrap it up with why it's really important. I've got just a couple of images of fireflies here. This is one of our local Massachusetts fireflies, Photinus ignitus, zoomed in pretty close. This is not zoomed in as closely, but this is actually a Southeast Asian Troptics firefly, another group of synchronously flashing fireflies that are out there. This is one from Thailand. And it's to remind me again that at the very end of this talk, I do want to, as I plug why this is all really important, take that even global perspective on it beyond just our local fireflies. And of course, the Massachusetts one is to remind me that to tell you that, yes, there is going to be a little bit of a New England bias to this, but I think we've tried pretty hard in Firefly Watch to make it pretty widely applicable no matter where you are. And of course, it's great to have Clyde giving a perspective for you guys that's a little bit south of you, and I'll give a perspective that's a little bit north of you. So some of the things I'll talk about you might not see, but many of them you probably would have a chance to see. So here's an outline of what I'll talk about. Some of this first part, identifying firefly genera and flash patterns, will be a little repetitive of what Clyde talked about, but I think it's worth reminding ourselves, because as we think about this problem, what we want you to do in Firefly Watch, go out and look at fireflies. That's sort of the first challenge, is knowing, is there any way to know what you're looking at out there? Then I'll talk about the actual observation procedure that we encourage people to do. And then even an opportunity to get more involved, what we call Firefly Watch Pro, which is a second tier of getting involved in observing these fireflies. So we'll jump into that. And again, we'll have some opportunities for questions as we work through this, similar to how Clyde structured things. So as we get started here, I am going to be focusing on 3 of the genera that Clyde mentioned to you: Photinus, Photuris, and Pyractomina. Now, I've left Phausis off this list in part because, as I mentioned, we don't really see them around here in Massachusetts. That's maybe my Massachusetts bias, but also I think more importantly because although we would love and encourage you in Firefly Watch to report if you're seeing Phausis, part of our protocol for looking at fireflies involves counting their flashes. And as you heard from Clyde, you can't actually count the flashes of Phausis because it continually glows. So that's one of the challenges with Phausis. But all 3 of these other genera are what we would think of as the flashing fireflies, who have different flash patterns. It's also valuable to talk about them as a group in this way because although we heard a little bit about how their morphology allows you to distinguish between them, there's also aspects of their flash patterns that can help you to distinguish between them. And that's some of the things that we ask people to report back on. So both what they look like this, but also what their flashes look like. In general, and this is, as I mentioned, a little bit of repetition, these 3 groups can be distinguished by some characteristics. Pyractomina, and maybe it's worth going back for a second, Pyractomina often have these darkened lateral margins on their elytra. One of the ways to set them apart from the 2 other genera. The other thing that we can see is that Photuris fireflies tend to have those long legs. Of course, they don't have that darkened lateral margin. In fact, you can see there's a lot of similarity between the pronotum of a Photinus and a Photuris firefly, where the Pyractomina is a little different from those 2. But as Clyde mentioned, the Photuris do sometimes, but not always, that's the other challenge that we encounter with these, have these stripes down their elytra. And so you can see there's the lighter stripe. What's harder to see is what he described before, the crease in the margin of the elytra that you can also see in Photuris. And it's also harder to see that they have, again, that more humpback shape, whereas a Photinus, and to some extent even a Paractamina, will have a flatter profile if you look at the side of it. So there's certainly challenges, but there are some things you can look for, and we encourage you to do that. Look for them, take pictures of them if you can, and we'll talk a little bit more about that in a moment. So some of the things to look for as you're trying to decide at least what genus of firefly I'm looking at. But as I mentioned, we also want you to look at their flash behavior. But before that, let me also point out a couple things about their lantern morphology that sometimes can be a clue in terms of finding them, if you have the good fortune of finding a female of one of these species. Males of these species are pretty similar in that they have lanterns that occupy these two segments of their abdomen. And so we can see that in males of Photinus, Photuris, and Paractamina. There's a Paractamina male, again, those same segments as a Photinus male. Photuris males, similar, although we actually don't have a picture of a Photuris male here because it is the Photuris females that are most famous for their predation. So again, we've got a nice picture from Lloyd of one of these predatory events. And the females do have, of Photuris, do have lanterns on both of those segments, but if you look carefully, the lanterns don't occupy the full segment. So that's another thing. And the Photuris females are the ones, if you're out there catching flying females, they're the ones you're most likely, the females you're most likely to catch, because the other females in Photinus and in Paractamina stay stationary. Clyde did a great job of describing this courtship behavior they're engaged in, where the males flash, the females stay perched in the vegetation and produce response flashes. And if you are able to key in on those flash interactions and find one of those females responding, often with a dimmer flash, because in Photinus, the female lantern occupies just a section of one of those two segments. And in Paractamina, the female lanterns occupy the margins of those two segments in many cases. And so that's another way you might be able to tell these genera apart if you're lucky enough to find a female. And again, with a Photuris, you're probably going to be lucky enough to find a female because they're out there actively hunting Photinus, but a little harder to find the Paractamina and Photinus females. So those are the differences in the lanterns. It's summarized here. There's a narrow band on a single segment in the Photinus females, wider bands across two segments in Photuris, and lateral bands on two segments in Paractamina. Now, the flashes themselves that are produced by those lanterns occur in a wide range of patterns that Clyde has given a good description of. This is a graphical description of it for a variety of species of Photinus. And so what we see here is the male flash on a timeline. Males flashing, for instance, every 3 seconds in Photinus marginalis, 4 seconds in Photinus sabulosus, and so on and so forth. And you can even see the double tap that Clyde had described for Photinus consanguineus, and see that it's not alone in producing those pairs of flashes that are repeated, Photinus carini, Photinus mcdermidi, and then even the Photinus carolinus that he described, a train of multiple pulses that, as he said, they synchronize. And then on that, using the same timeline, we represent when the females would respond. So a female might respond after just a half second after the male initiates his flash in something like marginalis. In some cases, female response delays are much longer. Photinus ignitus, females respond as much as 4 seconds after the male initiates the flash itself. And then in these multiple pulse species like consimulus and carolinus, the females can also have multiple pulses in their response. That's fairly rare. You can see even in these double flashing species, the females tend to have a single pulse in their response flash, but the females of consimulus and carolinus, consimulus is kind of a Midwestern species. Carolinus is that synchronous one that we find in the Appalachians. And those females do have, can have multiple pulses in their response. Now, So all of this is useful, and you can find this flash pattern information on the Firefly Watch website to kind of try to help you key in on what you might be looking at and figure out, is it a particular species, but also, again, it may help you to narrow it down to that question of what genus does it belong to. Because here's an example of Photinus pyralis. We have, I know I highlighted it. I highlighted it because this is the one that Clyde showed you the video of before, the Big Dipper, the one that as it produces this relatively, you can see it's got one of the longest duration flashes of all the flashes of these different species, and it produces it in kind of a J shape flying upward as it flashes. And I also highlighted, this is one we don't have in New England. Clyde has described it as one of the most, or I should say at least my part of New England, that there may be, I think there are some in Connecticut, but it's one of the most widespread fireflies out there. The ones they're incredibly hardy. They survive in mowed lawns and habitats that many other species don't survive in. And I just wanted to highlight it because one of the things that we're interested in Firefly Watch is asking questions like, is this spreading further north? Are we going to start seeing Photinus pyralis as a species that will begin to appear in places like Massachusetts where it hasn't in the past in response to changes like global warming? So it's a very interesting species, very widespread, very hardy. And of course, our other concern is that it might crowd out and outcompete for habitats some of the other species that would exist in relatively similar habitats, but wouldn't be as hardy and as tough a survivor as the Photinus pyralis, which are also relatively large for Photinus fireflies, probably one of the largest species of Photinus. So that's why I highlighted Photinus pyralis, something you're likely to see, especially in Maryland, where you are, probably the most likely Photinus you'll encounter. I've highlighted here an example of one of the Pyractomina. You heard Clyde describe them as often having amber flashes. That's not always the case, but they do. This is a timeline similar to the other one, but here we have the male flash just right above the female flash. So again, a way to tell them apart. The Pyractomina can have single pulses, or they can have very, very long pulses or trains of pulses in their flashes. And this is just a sampling of what is potentially out there in terms of Pyractomina. And also a quick example that we will be highlighting in a minute. Borealis is definitely one of the ones with the amber flashes. And I'll show you shortly just an animation we've created, or Firefly Watch created of that. Here's the variety that you might see in Photuris. And this is what Clyde sort of, said can be very, very frustrating and challenging because if you look at something like Photuris versicolor, you can't count on that it will have 6 pulses. Photuris versicolor is one of the large common predators on the other fireflies, so it may be exhibiting mimicry behavior when you capture it, but even within its mating, regular mating flash behavior, it may have multiple different pulses. And you can see that versicolor is not alone in the Photuris. Photuris, we don't have both male and female flash patterns up here. Oftentimes we don't have great information on what the female response patterns look like in these Photuris species. And often the females are producing their own, flashes very much like the male flashes. And they are all over the place in terms of what their flash patterns look like. So it's hard to generalize. I've tried to make a few brief generalizations here. The flash patterns and sometimes colors vary between species. Photinus tend to be more yellow. Pyractomina tend to, but you can't count on it, be amber. And Photuris often might produce slightly more greenish flashes. Those are a few of the things you could look for, but again, I wouldn't count on them as absolute ways to identify them. You really have to use them and these flash pattern charts in conjunction with the physical characteristics that Clyde already talked about and that I tried to highlight as well. And so as you piece those things together, we'll go back to the flashes for just a second. and just orient you again one last time to these ideas. We have a number of characteristics of a flash. If you have a single pulse species, what is the interval between flashes on average for the males? What is the response delay for the females? And what is the overall pulse duration for the males? Those are the things to look for. Ask yourself, how long is that flash in terms of its pulse duration? That's hard to measure because they're often less than a second, but is it about a half a second? Is it less? Can you make those kinds of comparisons? You can certainly count, as Clyde described, counting off once you see a male flash, how long till you see the next flash from what you think is that same male. Easier when populations are low. If you've got a field going crazy with fireflies, it becomes much, much harder. And if you can key in and watch a female response. Now, the one trick with watching female response delays is sometimes you can mimic the male flash with a pen light, especially as it's gotten later in the evening and male activity has started to die down. You may still find responsive females if you go out and you've been watching the male flash pattern and you've got a sense of what it is. You can produce with a pen light your own mimicked male flash and watch for female responses. And in that sense, see what that female response delay timing is. You can also watch females as they respond to males. Again, gets really, really hard when a field is just going crazy with fireflies, but easier earlier or later in the season. And often for many of these species, especially within Photinus, we will see males dominate the flashing early in the season, and you'll see then the sex ratio change as the season goes on and have many females that are still around towards the end of the season when there's not many flashing males, and that can be a good time to go out and do this kind of either observing females responding to the few males to get a sense of that female response delay, or also asking the question, can I simulate a male and get that female to respond and get a sense of that female response delay? So those are some of the ways we encourage you to figure out what you're looking at out there. And of course, there's one other piece of information. If you do happen to have a double pulse, as you heard, the double tap species, you could look at that between pulse interval. It can vary from species to species with either shorter or longer. So going back to Pyractomina, Pyractomina borealis, often out very early in the season. And Clyde mentioned a number of the Pyractomina tend to be out earlier in the season than the Photinus, for instance. And as they come out, the borealis are often found in trees. And here's just a brief animation of that flash pattern. It's meant to look kind of amber. I'm not sure that it really does, but again, a single pulse repeated. And you can imagine counting off 1 Mississippi, 2 Mississippi, 3 Mississippi, 1 Mississippi, 2 Mississippi, 3 Mississippi, 1 Mississippi, 2 Mississippi, 3 Mississippi, to try to just, to reconstruct that you've got something flashing every, 2 to 3 seconds, somewhere between 2 and 3 seconds there, to potentially identify it as a Pyractomina borealis. Of course, again, it helps to catch it and then see that it has those lateral margins on the pronotum that would let you know that it's a Pyractomina, as opposed to something like a Photinus. So Pyractomina produce single pulses or flickering flashes, and the different species can be distinguished by their duration and their flash pattern intervals. Back to the Photinus again. So you can imagine if you got something flashing every 2 to 3 seconds, it could also be Photinus colostrands here. If you've got something flashing every 4 or 5 seconds, it could be Photinus ignitus. And so, once again, you can, Photinus ignitus, you wouldn't find in the trees like those Pyractomina, you'd find it in more in fields. And here's another representation of this process you might go through counting a flash. So 1 Mississippi, 2 Mississippi, 3 Mississippi, 4 Mississippi, 5 Mississippi, 6 Mississippi, 1 Mississippi, 2 Mississippi, 3 Mississippi, 4 Mississippi, 5 Mississippi, 6 Mississippi, and see those flashes repeating about every 5 to 6 seconds, as we see represented for Photinus ignitus up here. I've moved on with the highlight to Photinus green eye because we have one more or one other Photinus animation for you of those green eye. They're often found in fields very much like Photinus ignitus are found in, but they do produce those double pulses. Let's see, can I get it to do it? Maybe I'm just not waiting long enough. Let's go back for a second. Back. That should do it. See if our greeni appears. There it is, 1, 2, and then a pause, and 1 Mississippi, 2 Mississippi. So flashing about every 1 to 2 seconds. And then you can also count the pauses between those pairs of flashes. So this is just another representation. And I think we threw in a female response there too, for you down in the vegetation. So anyway, Photinus males produce these single, double, or again, as you saw in the case of Photinus consimilis and Photinus carolinus, multiple pulse flash patterns that, along with the female response delay, you can also try to use to identify down to the species. And finally, we'll show you a quick animation of Photuris versicolor we've created. There they are producing those trains of multiple flashes. You can find Photuris fireflies flying up high in trees. You can find them flying down low, especially when they're hunting Photinus. And they come, as you've already seen, frontalis's pattern. That's an example of versicolor's pattern. There's the frontalis that Clyde talked about, that actually synchronizes, just producing those single pulses about every second. Very different from the Photuris versicolor. So I hope that gives you a little bit of an idea of what you can go out there. And again, they produce a wide range of flashes, and they sometimes mimic Photinus. So again, best to catch one and add that information about whether you're looking at the genus Photuris based on those long legs, that more hunched body, and as we mentioned, in oftentimes a larger size, and in some cases also that stripe down the elytra. So are there any questions? Do people think they have some clue of beginning the process of going out there and looking at fireflies? Do they have any questions about things they've seen out there in the field?
Lynn has raised her hand.
Okay, I just was curious. Some of them are earlier in the evening and others are later at night. Is there any listing that shows those differences? Because I think that makes it easier too, right?
Yeah, that's a good point. And I think on our Firefly Watch website, I think we do have some input about that. Again, Clyde's already mentioned the one that, that's going to come immediately to mind, which is Photinus pyralis, that really common single pulse flashing that has the sort of J shape to its flash, the Big Dipper. That is a very early evening flasher. And as a result, as he described it, you can actually see the silhouette of its body as it's making that flash. If you're looking sort of up at it, and you've got the light of the, sort of the setting sun, behind it, or at least the twilight behind it. Whereas other species, you're not going to see them until it's too dark to actually detect their body against the background. I showed you another example. Photinus greeni is another species that flies very early in the evening, whereas the other example, Photinus ignitus, I showed late at night. I guess my main point, and Clyde may offer something else about this, but in general, the early flight is the exception rather than the rule. And so when we point to things like Photinus pyralis and Photinus greeni, they are unusual in being early flyers, and that does help you to identify them. I don't know if Clyde, if you add anything or other examples.
Well, I'd add that some of them, a great many of them, don't start until it's flat dark, and sometimes particular species won't start until sometime after it's flat dark. And then the other important thing to note is that some of them will flash for a very brief period of the time in the evening. So the consanguineus, the double tap, a lot of times they'll be out courting. The males will be flying around, and they'll start later than the Photuris, but not necessarily when it's full dark. But they only fly for 25 or 30 minutes in the evening, and they're done until the next night. And so that's another issue that you need to take into account, is that in order to detect all the species that might be in your area, you need to be out more than just one part of the evening. So, and by the time pyralis is kind of slowing down, lots of others are kicking in. And then some of them, like frontalis that I have in my yard, it's 1 o'clock in the morning and they're still out there trying. Those guys have heart. They never give up, and they're still flashing in the middle of the night. So, you know, some of them will flash for a very brief period in the evening, and some of them will flash for hours in the evening. Did that help?
All right, Tom, I think so. Tom asked, are flash intervals temperature dependent?
Thank you for asking that, Tom, because I was supposed to say that and I didn't. So they absolutely are temperature dependent. So all of those time elements that I described, and that's why I often said 5 to 6 seconds and so forth, as it gets colder, all of the durations are going to become longer. So whether you have if you have a species producing pairs of flashes that are repeated at a particular interval, both the time between those pairs is going to become longer, and the time between between those intervals is going to become longer, and even the apparent duration of the flash is going to appear to become longer. So all of those elements become longer as it gets colder and become shorter, in contrast when it becomes warmer. So we do ask, as part of Firefly Watch, that people record the temperature and report the temperature that they're observing at, because that can be helpful in figuring out what you're looking at. And in general, the flash charts that we provide through Firefly Watch do note what temperature we're sort of thinking of as the standard temperature, so that you can then, in your mind, adjust downward or upward if you're in much hotter temperatures or in cooler temperatures. And then, of course, there are temperatures where it gets down so much that they won't fly at all, and sometimes even where they won't flash at all. So that's the other impact of temperature, in addition to on the flash patterns themselves.
And Jerry wanted to know, is light pollution detrimental?
So yes, and this is a small but gradually growing body of literature out there on the negative impacts of light pollution on fireflies. In fact, there's there's new stuff coming out all the time about it, but the evidence so far, in a variety of ways, is showing us that yes, this, these flash behavior is going to be impacted by light pollution. Now, this connects back to, I think it was Lynn's question, right, about when species fly, because and Clyde's follow-up information about that. Some species are going to be less impacted by light pollution. So I described Photinus pyralis as a hardy species in terms of its the habitats it can live in, living in things like even manicured lawns that would not support most firefly species because it's just too dry a habitat. But another factor about Photinus pyralis is, as an early evening flyer, they may be less susceptible to things like light pollution than other species. So it's a fascinating, growing body of literature, but it really is an issue and something that we continue to look at. We have there's a lot of ongoing projects, hopefully continuing to document that. And I think I heard that you guys are going to have someone in, a month or two talking about light pollution as well, in one of your talks. I think we heard that in the intro here.
I'd add to that, even Photinus pyralis behavior can be altered by artificial lights, because you may notice if you have a street light or a porch light, that sometimes they'll continue calling much later into the evening than they would, because the ambient light still mimicking dusk. And one other thing about lights is that lots of fireflies are actually attracted to artificial lights, which, I mean, if they're wandering around your porch light, they're not out doing what they're supposed to be doing. And if they're wandering around your porch light, it might be a easy opportunity for a Photuris to come nab them too. So many of them are oddly attracted to artificial light, and that can mess up what they're supposed to be doing. And bear in mind, most adult fireflies only live a couple weeks. They feed for the most part, as adults. And so they have a pretty limited time to accomplish their primary mission in life, which is to reproduce.
All right, I think we can keep going.
Okay, excellent. So moving on now, let's talk about Firefly Watch itself, talk about how you can get involved, what we're trying to accomplish here. And so this is just the entry page. If you Google Firefly Watch, you'll get to this Mass Audubon site. Oops, it's moving itself along. I don't think there's too much to show here. I'll be putting up the email contact again later. I'll be putting up the URL again later. But you can see there's a fair amount of information here about getting started, how you submit observations. You can see the data that we're working from. I'll keep learn a little bit about some of us who are researching fireflies, some of the facts about fireflies, and of course the resources like those flash pattern charts that you can use in order to try to figure out what you're looking at. Okay, so how to join? The important thing is that we want you to identify a habitat. We want you to keep going back to that habitat and to describe it, and ideally go back to it year after year. We're trying to really get, the goal here is to get longitudinal data in those chosen habitats, to note the date, time, and weather data, as I mentioned, the temperature, but also precipitation, because that can impact firefly activity. And then for 3 10-second intervals to count, sorry, that's going a little faster than I want to count the number of flashes seen. And this is why I said Phausis you can't do that for Phausis, but you can do it for these 3 other genera, to do these kinds of counts to give us a sense of the abundance of the fireflies you're looking at, and then you input your data into the Firefly Watch system. That's the general pattern of what we're asking people to do in our standard Firefly Watch. Now, you'll notice that other than submitting your own personal information to us about what species or genus you think you're looking at, we're not actually absolutely concerned about that. We're just, with this level of Firefly Watch participation, interested in getting some general ideas about, are you seeing them, and when are you seeing them? We hope we can get information about when they first appear in a particular part of the country, and get a sense of when they disappear in that part of the country, and get a sense of generally what the abundance is like, using those 3 10-second intervals where you count the number of flashes seen. Now, as Clyde pointed out, picking a time of night to do this is pretty critical, because they are going to, a particular area you're looking at in particular species are going to come out at different times. And so if you're interested in diving more deeply into those differences between species at your particular site, we do have another opportunity that we call Firefly Watch Pro. So here's a quick summary of some of the differences in Firefly Watch. We ask you to go out 1 time a week, provide your location coordinates, provide the habitat type, mowing, weather, artificial light. We're looking for those factors that might be impacting the firefly abundance really broadly, and do those total number of flashes 3 times, 10 seconds each. We actually also ask you to tell us something about the total number of different flash patterns. So that's where it is useful to think about, am I seeing any double flashers? Am I seeing single flashers? Am I seeing multiple pulses? Those kinds of things I showed you. We do ask you to report that in Firefly Watch too, so we can get a little bit of a sense of the species diversity that you might have at your site. But again, no efforts to really hone in on what genus or species you're looking at, unless you want to get into Firefly Watch Pro, which we've just been doing for a couple of years now. The Firefly Watch in general, we've been doing, I think, for over 10 years now, collecting this kind of longitudinal data. But Firefly Watch Pro is a new initiative where we ask you to go twice a week. Again, provide your location coordinates, but also we ask people, and this is not a bad idea for Firefly Watch as well, but we ask you to actually tell us exactly the direction you're looking in, so that you go back and look at exactly the same direction each night, and you look at exactly the same area. Give us an estimate of the spatial area you're looking at, because again, that can be a challenge. As people have done Firefly Watch, that sometimes they say, oh, in that 10 seconds, I can't even count the number of flashes I see. There's so many of them. And so you can, in Firefly Watch Pro, sort of identify an area of a field or an area of a forest. Say, I'm just going to look at this area and count them within that area, which can give you a better sense. So again, a lot of the same information, although we've added a few other potential disturbances like fire ants and other disturbances that you can, and pesticide use for you to report in Firefly Watch Pro. And then what we're asking you to do in Firefly Watch Pro is actually go 3 times in that same night, spread out about an hour apart. So capturing what Clyde was describing in terms of the diversity that might occur across the course of a night. And again, tell us the number of flash patterns. And then if you can find identify what you think is a particular species, tell us about that one species number of flash patterns on those 3, a number of flashes on those 3 instances, so that we're getting data not just about flashing in general, but ideally data about what's happening to a particular population of a particular species, if you think you can identify one down to species. I see we have a question here, and I maybe we should capture it right now while it's fresh. Lynn, you want to go ahead and ask your question? You're muted still.
Sorry about that. Now I got to remember what it was, but I wrote it in the chat. I noticed a marked increase in the number of fireflies replacing the lawn with natives, but how likely is it that new species would move into the area? Are they long distance flyers, or am I just seeing more of the same species that were already here?
So the answer, I guess, depends in part on the genus you're talking about. You heard me describe for Photinus and Pyractomina that the females are very sedentary. And of course, the females are the only ones that are going to be laying eggs and contributing to the spread of a species geographically. So for Photinus and Pyractomina, we think that their spread is going to be pretty slow. And again, the larvae, there's only so far they're going to crawl as well. So I think for those 2 genera, you're most likely to see the same species just spreading out and occupying more space and becoming more abundant, which is a great thing. There is some possibility for Photuris, because the females are much more active and are flying around, that you might see Photuris species spread a little bit more widely and come in and invade areas that they wouldn't otherwise. But I'll let Clyde also comment on what he thinks about that.
Yeah, so with the Photinus, it depends a lot on the species. So several of the species of Photinus have females that are wingless, larva form, and they can't disperse any further than those females can crawl. But when you replace a lawn with native vegetation, you're probably changing the microclimate so that that habitat now is more desirable, more supportive of fireflies. If you've got taller vegetation, it's probably moisture at the ground level, and so what you're probably seeing is mostly an increase of species that were already present but at lower levels.
Yeah, and I will just echo that with one other perspective, because it's even females that are fully capable of flight. I've done surveys of Photinus ignitus females where I would catch one, mark her with paint, put a flag down, and I could come and watch the same female in the same spot night after night after night. They do not travel far. So it is a challenge in terms of their what we see as the potential threat to them of habitat disturbance, and it's why it's really valuable to preserve those habitats, and as Clyde described, particularly preserve moist soil conditions, native plants, all of the things that you're doing. So that's great.
So yeah, if you look at it from that, if you look at it from that female firefly's perspective, if she's already in a good spot, it's really risky to try and find a better spot, right? And so they don't move a lot. He's right.
Yeah, and and again, the other thing to remember is, unlike other species where the females have a reason to move for foraging, fireflies don't have that. So they're sort of they're different in that respect, and as again much more susceptible to a population just being completely wiped out by things we might do to that particular habitat, because they're not going to go someplace else. So, and particularly the time of year when all of the larvae are in the soil, there's no adults around at all, then a lot of our, housing development practices that involve turning over everything with a backhoe before we, go and put up a bunch, one or multiple houses, are just that population is most likely gone, because all of those larvae have then been buried deep into soil that they can't survive. So anyway, those are some things to think about, and why we're interested in tracking these populations and seeing what's happening to them. I'm going to tell you a little bit more about Firefly Watch Pro before we turn it over to more questions and kind of wrap up. And so Firefly Watch Pro uses a different portal than Firefly Watch. It uses something called Anecdata, a system designed for entering community science, designed for community science projects like this. And so this is where you would, if you're using a laptop, where you would be entering information about your location and yourself and the date, and as I mentioned, the cardinal direction. You would then, enter information about the focal area, how big an area you're looking at. You'd provide us an email so that we could follow up with you. Here's the information about things like pesticide use, light pollution, so forth. And then here's that question of how many flash patterns are you seeing across those 3 time observation periods? What is the time that you're making the observation, the number of different flash patterns you're seeing, so we can see something about the diversity. And then here's where you would make individual species observations if you think you could find a specific flash pattern to watch, again, across those 3 time periods, and observe, what is the between pulse interval, what is the flash pattern interval, and how many flashes are you seeing of that particular type in that time frame. And finally, you can see down the bottom, it says add a photo, another thing that we ask you to do in Firefly Watch Pro that isn't part of regular Firefly Watch. We also really encourage people involved in both projects to upload their photos to iNaturalist. There's an excellent and growing Firefly community on iNaturalist, where you can get help in identifying what genus and possibly in some cases even species you might be looking at. So that's really a great thing to do. In addition to this add a photo is part of the Anecdata. It doesn't go to iNaturalist in that case. We still want you to do it, but once you have that photo, please also, we encourage you to add it to iNaturalist as well. And then finally, should note that the other nice thing about the Firefly Watch Pro is that you can do it all on your phone. These are screen grabs from my phone. There is a mobile app for Anecdata, and where you provide all of that same information. Here's the information about those number of flash patterns that you could record over those 3 time periods. And here's the overall interface, what it looks like when you log in on your phone, all this information. So I won't belabor you with it. And of course, iNaturalist also has an app for that as well. So you can add your information that way. All right, we're back to questions, and I see Lynn has one, but maybe also there's some in the chat. So I'll let our moderator let us know what there is.
Yeah, as the moderator, I have my own question. And so you're not asking people to actually collect the insects and take a look to get more species and information and take photos of that, or when you're looking at the pattern, if you're trying to get used to counting the variations of the patterns and the frequency, do you recommend people video that and then take it back and try to look at it? I mean, if you're just going out there, it's kind of a whole hodgepodge unless you have those visual clues in your brain, a search engine already built into your brain.
Yeah, it's challenging. So we do encourage videoing, and you can upload video to iNaturalist as well. And I know those of us in the Firefly community, there's been some conversations going back and forth, about increasing the use of video in iNaturalist. Although Clyde's videos also illustrate that it is a little hard to get really, really good quality video as well of these flashing lights. So, but we encourage trying it. We encourage a catch and release approach to fireflies, as opposed to the catch and smear all over your body approach. So yeah, catch, because to get a good photograph of it, it can be very challenging unless you've caught one. And so we don't discourage catching them for the purposes of getting a photograph, but we do encourage then releasing them back into the same habitat, and using the photo as the way to make identifications. We're not asking people to send in samples. Now, there are in some parts of the country efforts. There's a Western Firefly project in Utah and New Mexico. Again, they're not asking people to send in samples, but they are, in that case, actually sending out experts to go, and in some cases, those experts will collect the fireflies and bring them back to the lab to identify. And we may move to some of that activity as kind of a next layer of Firefly Watch, particularly as we begin to identify species that are really threatened. And that's why it's going on with the Western fireflies. There's a very small number of species out there, and they're in very small pockets, and we think some of those species are threatened with extinction. And so that's the justification there. But we think that's going to be true for some species of Eastern fireflies as we learn more about them. Some we think will be around for a long time, like Photinus pyralis, but others of the many species you're hearing about may be threatened. And we, as we start to identify that using data from experts, but also data from Firefly Watch Pro, we might take a step where we'd be collecting. But we don't ask anybody else to collect and preserve specimens, because if we were doing that on a large scale, that would have its own threat to firefly populations. In fact, there's a history where not only were they being collected to smear on people's bodies, but for biochemistry, they were being collected to get, sorry, that's my dogs, to get the luciferin and luciferase, and we do believe that it has had, and certainly has the potential to have, negative impact on firefly populations.
I'd add to that, if you're taking pictures of fireflies for identification purposes, it's ideal to get a good picture of the dorsal side, the back, and it's really important sometimes to get a good picture of the underside, because sometimes we can't distinguish species without a good look at the belly, basically.
Yeah, and as you recall, if you have a female, that can allow us to identify it down to the genus level, at least, having that belly view, what we call the ventral view, and then we saw all the other characteristics of the dorsal view that are useful, the back view. So yeah, both, please, that would be great.
All right. And Laura's interested in learning more about versicolor complex.
Yeah, so the versicolor complex, we have a couple species actually that are that have been for a long time sort of just like taxonomic dumping boxes, where they look sort of the same, they act sort of the same, so we're not really sure if they're not the same. And versicolor is one of those that is actually, versicolor is actually probably, it could be as many as a dozen different species or more. It's just that we really don't have the tools or the data yet to conclusively identify the species within that complex. And so when we say the versicolor complex, a lot of folks will say this is Photinus versicolor, but it could be Photinus special versicolor of a different species. We really can't tell yet. And so that's where that comes from. And that's not the only species that's probably a complex of multiple species that just happen to look very similar. In a lot of insects, that are, you can take in most insects, you can take a genus of insects that look very, very similar to the naked eye and look at the genitalia of the males, and you'll find that they're dramatically different. That's not the case with lots of fireflies. And so what happens, we think, is with fireflies is that they actually segregate into different species based on their behavior more than changes in their morphology. And so that's a problem. In fact, there's not a lot of variation in the genitalia in Photuris, period. From well-recognized species, a lot of times the genitalia look fairly similar. And so we don't have the same kinds of tools. And you can do genetic work, but you have to have some understanding of what the genetic variation within a species is before you can identify between species. And if you're dealing with something like versicolor, how do you start? So I hopefully that just muddied the water completely.
No, thank you.
Yes, the Photuris are uniquely challenging because of the combination of not having the consistent flash patterns and having these issues of morphology that don't allow you to distinguish them. I mean, by contrast, it is pretty good if you have Photinus males. You can dissect those apart and tell them apart through the male reproductive organs, but not so much for Photuris. Other questions?
Are there any other questions?
I have a question. I just was curious, when you do the iNaturalist, do you have a problem if we obscure the location? This would be obviously my backyard. Is it just as useful if it's obscured, or do you really need to pinpoint the exact location?
So it certainly is helpful. But again, it's helpful in terms of general geographic information. So, if you want to sort of, try to add notes that just say, this is the general area, that might be a nice compromise, because we certainly don't want anybody being uncomfortable with it. As you know, when you're reporting to Firefly Watch itself, we are asking for, things like coordinates, but when you're putting something in iNaturalist, that's a different boat. And so I would say you don't have to, if you don't feel comfortable with it, because that's a different group of people looking at it, a broader group of people. And yeah, I wouldn't want anybody getting uncomfortable, but it might be worth noting the state you're in, even just because we know that, again, these fireflies do have ranges, and that can help us to help whoever's looking at it to determine more carefully what it is.
My understanding is that when you obscure it, it just makes the radius of where the location.
Oh, okay, good. Yeah, then that's great. That would be perfect then. I'm actually not one of the people who looks at and tries to help identify them through iNaturalist. So.
In terms of the longitudinal study, in terms of status and trends, and when you talk about the unlikeliness of the populations to migrate over time, very vast distances, so that if a population is wiped out in one habitat, it's wiped out. Are there any repopulation efforts in different areas where there used to be fireflies and there aren't anymore, or?
So a lot of people are doing what Lynn was describing, certainly trying to make their habitats more firefly friendly. I think I have talked to one or two people who, have, caught fireflies in one place and tried to transplant them to another, but I don't know of any major efforts to do that, except in Asia. In Asia, where there is this incredible cultural tradition of firefly festivals, and they think of, historically have thought of fireflies as the spirits of their ancestors. There are scientists who work with communities to create firefly habitats and then actually introduce fireflies into those habitats. So it's not completely far-fetched. People are doing it. It's just not something that I see us doing around here. And it's certainly, at least in terms of our data so far through Firefly Watch, it's not at the point of being a priority, because the bigger priority is, again, the kind of thing Lynn's doing of saving the habitats that we have and making sure that the habitats we have are preserved in a way that's appropriate for fireflies. But we are also just at the beginning. We over the past year, we did our first North American, what's called red listing process. This is the International Union for the Conservation of Nature, has a, and see, I may even have a slide about that. Yeah, here's the IUCN Firefly group. They have a process. So when you hear about species being identified as, threatened or endangered, certainly there's a federal process for that, but this is an international process that's pretty widely respected for identifying species as at risk, putting them into different levels of threat. And we've just gone through that kind of assessment for North American fireflies for the first time. One of the categories they can be put into is data deficient. And I think, many of our fireflies might fall into that category, although many of them, like something like Photinus pyralis, we would never consider threatened because you can find them so many different places. But as we do that, I guess my main point is, as we do that, that might open the window to questions about whether some of these populations need to not just be protected, but even even be introduced into new areas. Now, we're not at that point yet, but we are getting there. And so it's a fascinating question.
And by the way, it's important to remember that you can't move a population of fireflies unless you're moving the females. You can catch all the males you want to and turn them loose, and they'll just wander around, love lost and forlorn. So you got to move the females. Yeah.
All right, any other questions out there? This has been a phenomenal presentation. Thank you so much. I can't wait to get out there and now understand better what I'm looking at in my backyard and teach other people and get involved with Firefly Watch. So we're all going to start talking about it and recruiting folks, because all data is good data, and we'll get that in there and try to get more Maryland on the map.
And
you'll be stunned. Go ahead. I'm sorry, Chris.
Oh, I just want to put in a couple more plugs, other places for information. As you can guess from the fact that it's the International Union for the Conservation of Nature, these firefly efforts, although we started with North America, are something that we're interested in doing worldwide. And there's another organization, Fireflies International Network, that is a partner in these efforts and has its own website you can check out. And then here's just a whole list of those websites, including the Mass Audubon, including the Anecdata, the iNaturalist, the email for the Mass Audubon Firefly Watch to get involved, my email if you want to go and do Firefly Watch Pro, and the IUCN and Fireflies International website. So throwing those up there for the end of the show, and I'll let Clyde take it away.
No, I was just going to say it's something that's really, really fun and fascinating to get involved in. I came into them kind of late in my career, and I'm just floored by the diversity of fireflies. I have at least a dozen species in my yard, and that's it's really exceptional. You probably do too.
Yeah, that's amazing. I mean, again, what's out there is when you start looking, you find stuff. And so I want everybody to stay curious and get out there and start looking. Again, thank you so much, Clyde and Chris. And we'll we will definitely stay in touch and hopefully provide you with some great data from Maryland.
Find
Excellent.
us some, they're there.
Yeah.
We're going to be fine. We're going to be looking for you. All right, everybody, stay safe, stay curious, stay outside, and stay well, everyone. Stay safe.
Thank you.
Take care.
Thanks,
See you
everybody.
soon.
Thank you.
Bye-bye.
Bye.